Dear students, today we are going to discuss on the topic metabolism. The metabolism of an organism may be defined as the sum total of all the enzyme catalyzed reactions that convert nutrients into energy and complex chemical finished products of cells that occur in organisms. It is a Greek word meaning change. So how can we define metabolism? It is actually divided into two pathways. One is anabolic pathways and other one is catabolic pathways. Anabolic pathways will be involving in the synthesis of the complex chemical finished products of cells. And catabolic pathways will be involved in the conversion of nutrients into energy. So both combined together is called as metabolism. And coming to the types of metabolism, as we have discussed earlier, anabolic pathways, catabolic pathways, and one more type, that is amphibolic pathways, also come under types of metabolism. Anabolic pathways are those involved in the synthesis of compounds. Protein synthesis is such a pathway, as is the synthesis of fuel reserves of triacylglycerol, otherwise called as triglycerides, and glycogen. Anabolic pathways are always endergonic because we have to put some energy for the synthesis of bonds present in the chemical biomolecules. Catabolic pathways are involved in the breakdown of larger molecules into simpler substances. Usually catabolic pathways are exergonic, reducing equivalents, and mainly via the respiratory chain, and forms ATP. This ATP is usually formed as a result of catabolic pathways. Amphibolic pathways occur at the crossroads of metabolism. That is, it is acting as a link between anabolic and catabolic pathways. We can say citric acid cycle as an example. How can we outline the pathways of metabolism? So whenever we take food, it will be composed of usually carbohydrates, proteins, lipids as macronutrients, and vitamins, minerals as micronutrients. The macronutrients such as carbohydrate, protein, and fats will be initially digested in the mouth. The digestion actually starts in the mouth with the help of saliva, and the teeth will be masticating the food materials along with saliva and convert those food materials into bolus. This bolus will be swallowed by us and it enters the stomach via esophagus or food pipe. The stomach will be involving in the digestion process by secreting hydrochloric acid. The carbohydrate digestion will be occurring in the duodenum with the help of pancreatic amylase, and the proteolytic enzymes will be secreted in the gastric part and it will be helpful in the digestion of proteins, and fats will be digested in the small intestine or duodenum with the help of bile secreted by the liver. Finally, after digestion and absorption, carbohydrates will be converted to simple sugars, proteins into amino acids, and fats into fatty acids and glycerol. And all those simple nutrients will be catabolized to form a common central molecule called acetyl-CoA. This acetyl-CoA is formed as a result of catabolism of simple nutrients for the provision of energy in the form of ATP. This acetyl-CoA will be entering into citric acid cycle where reducing equivalents are formed, and those reducing equivalents will be entering into electron transport chain and finally forms ATP. If not needed, if energy is not needed, if it is present in ample amounts in the cells will be deciding in the storage of those simple nutrients into complex molecules. For example, the glucose can be stored in the form of glycogen in liver and skeletal muscle, and the excess fatty acids can be stored in the form of triglycerides in adipose tissue, and amino acids will be never stored, and the tissue proteins will be broken down to amino acids as if needed, and amino acids can be converted back to tissue proteins. The next slide is about the overview of carbohydrate metabolism. Coming to the overview of carbohydrate metabolism, we can obtain glucose after digestion and absorption of carbohydrate food materials, and this glucose can be converted to pyruvate, and pyruvate can be oxidatively decarboxylated to form acetyl-CoA, and this acetyl-CoA can enter into citric acid cycle to form reducing equivalents, and reducing equivalents will be entering into electron transport chain for the formation of ATPs. This is a major route of oxidation of glucose for the provision of energy. If suppose energy is not needed, this glucose can be stored in the form of glycogen through a process called glycogenesis. If at all it is needed between meals, the glycogen stored in the liver will be released via the process glycogenolysis and converted to glucose 6-phosphate, and glucose 6-phosphate will be entering into glycolytic pathway, and it forms energy through electron transport chain. The glucose 6-phosphate can also be oxidized through an alternative route called pentose phosphate pathway or HMP shunt, where ribose phosphates are formed and NADPH molecules are also formed as end products of pentose phosphate pathway. These ribose residues will be helpful in the formation of nucleotides and nucleic acids, and NADPH will be mainly involved in the synthesis of fatty acids. The other alternative route of glucose 6-phosphate is uronic acid pathway, where this glucose 6-phosphate will be converted to glucuronic acid. This glucuronic acid will be acting as a conjugate in detoxification reaction, and it is also playing an important role as an ingredient in the formation of mucopolysaccharides. And the triose phosphates formed as a result of glycolysis will be combining with fatty acids to form triglycerides, and the acetyl-CoA, which is formed as the end product of pyruvate oxidation, will also be involved as a precursor of cholesterol biosynthesis, and the pyruvate can also be converted to amino acids, and this pyruvate can at times be helpful in the formation of glucose through gluconeogenesis pathway. This is all about the overview of carbohydrate metabolism. Now we discuss about the overview of protein metabolism. Proteins after digestion and absorption will be converted to amino acids, and amino acids will be classified into essential and non-essential amino acids. Essential amino acids are normally synthesized, that is, essential amino acids must be supplied in the diet since they are not synthesized by the tissues. And non-essential amino acids can be supplied from the diet, and those non-essential amino acids which are also formed from the intermediates of citric acid cycle by transamination reaction, and excess amino nitrogen as a result of deamination of amino acids is removed as urea, and the carbon skeletons that remain after transamination give the following products, that is carbon dioxide and water, glucose by gluconeogenesis, and ketone bodies. The amino acids are also the precursors of many other important compounds, for example purines, pyrimidines, and hormones such as epinephrine and thyroxine. Coming to the overview of fatty acid metabolism, the long chain fatty acids are synthesized from acetyl-CoA derived from carbohydrate or from dietary lipids. In the tissues, fatty acids are oxidized to acetyl-CoA to form fat, which is the main caloric reserve of the body. Acetyl-CoA formed by beta oxidation has the following significant roles, that is, It liberates carbon dioxide and water and also yields high energy. Therefore, during the oxidation of fatty acids by beta oxidation, for a complete oxidation, more energy is needed. And this acetyl-CoA is also the source of cholesterol biosynthesis. In the liver, it forms ketone bodies, which are alternative water-soluble tissue fuels. These fuels become important sources of energy under certain conditions, for example, starvation. And the location of metabolic pathways can be classified into two types, two major groups. One is at the tissue and organ level, and other one is at the subcellular level. At the tissue and organ level, the liver takes up excess glucose and converts it into glycogen or to fat. During meals, it causes glycogenolysis to supply glucose in the blood, or in company with the kidney, it converts non-carbohydrate metabolites such as lactate, glycerol, and amino acids to glucose. Skeletal muscle uses glucose as a fuel, forming lactate and carbon dioxide. It stores glycogen for its use in muscular contraction and synthesizes muscle protein from plasma amino acids. When we discuss about intermediary metabolism at the subcellular level, mitochondrion acts as the focus and crossroad of carbohydrate, lipid, and amino acid metabolism. Particularly, it is the house of the enzymes of citric acid cycle, of the respiratory chain, and beta oxidation of fatty acids and ketone body production. It is also the collecting chamber for the carbon skeletons of amino acids after transamination, and for utilizing these skeletons for the synthesis of non-essential amino acids. In the cytosol, glycolysis, HMP shunt, and fatty acid synthesis all take place. The membranes of endoplasmic reticulum contain the enzyme system for acyl glycerol synthesis, and the ribosomes are responsible for protein synthesis. Finally, we enter into the topic integration of metabolism. Pyruvate is converted to acetyl-CoA, which is the starting material for the synthesis of long-chain fatty acids, and by the reverse process, fatty acids are converted to glucose, whereas the pyruvate dehydrogenase reaction is essentially non-reversible, which prevents the direct conversion of acetyl-CoA to oxaloacetate via the citric acid cycle. Since one molecule of oxaloacetate is required to condense with acetyl-CoA, and only one molecule of oxaloacetate is regenerated. On the basis of similar reasons, there cannot be a net conversion of fatty acids having an even number of carbon atoms to glucose or glycogen. Only the terminal 3-carbon portion of the fatty acid having an odd number of carbon atoms is glucogenic, as this portion of the molecule will finally form propionyl-CoA by beta oxidation. Oxaloacetate is an intermediate both in the citric acid cycle and in the pathway of gluconeogenesis. Many of the carbon skeletons of the non-essential amino acids can be produced from carbohydrate through the citric acid cycle and transamination. Glucogenic amino acids can also produce carbon skeletons that are precursors or members of the citric acid cycle by the reversal of these processes. The ketogenic amino acids are converted to acetoacetate, which is metabolized as ketone bodies, forming acetyl-CoA in extrahepatic tissues. Hence, under starvation, a net breakdown of proteins and amino acids is accompanied by a net breakdown of fat. The net conversion of amino acids to fat is not a significant process except in animals receiving a high protein diet. So let us meet together in another interesting topic. Thank you.